Hi, welcome to our Cell Theory Day 2, which is Cell Theory and Life 101 if you're looking in your notes. So today our warm-up questions are on the screen right now. How do we find the total magnification of an image that we are observing through a microscope? Well, we're going to multiply the magnification of the eyepiece times the objective lens magnification. So in our classroom, our eyepiece is always 10, and then our low objective is 4, our medium objective is 10, and our high objective is 40. So you would multiply 10 times whatever the number is of the other lens that you're using. You're always going to start on low power, and you're always going to start with your microscope stage all the way down whenever you're viewing slides. And once you've focused on low power with your coarse adjustment knob and you are ready to move to medium power, you know that you are observing something at 100 times its normal size because we have eyepiece is 10 and the medium power objective lens is 10. So 10 times 10 is going to be 100. Okay, so today in class, I gave everybody their new blue lab notebooks. This is your scientist notebook that we're mostly going to use for third quarter. And as I handed these out, I asked people to write first and last names on their books, right on the front cover, the period number, and Thomas, as your science teacher. We don't have extras of these. We have one for every student, and so you need to keep track of this. Bring it back and forth to class every day because sometimes I'll have you turn it in so that I can do a quick grade. But you want to bring it back and forth every day and keep track of it, please. All right, so in class today, we practiced microscope skills and scientific drawing skills. That's because we want to make sure we can use the microscope and focus on the cells so that we can see not only the cells, but also their little parts inside. That's the organelles. All right, we had a quick quiz on microscope parts in the classroom, and so if you missed class, be sure to come and check that out. Get a makeup quiz. All right, so the quiz, I pointed to different components of the microscope and asked everybody to fill in the quiz as I went around the room. All right, so here we have our self-assessment scientific drawing checklist so that we know what to do when we're drawing a scientific drawing. So we are going to, I'm not going to give you a whole minute, I'm just going to talk. When you are drawing things, you're going to especially draw the parts that are requested by your teacher or your assignment. You want to draw your cells only using pencil. Okay, the drawing is going to use up the appropriate amount of space. So if your view that you're looking at needs to show the entire view, you might fill up this entire circle. Most of the time, though, I'm going to show you an example. If I have cells all over my slide, usually we'll choose 8 to 10 cells to draw, and we'll just label on one of them. It doesn't make sense to have to draw, 87 different cells when they're pretty much all the same. All right, but we do want to try to have the same proportional size when we draw it compared to what it looks like as we're looking in the microscope. Okay, the drawing's going to show attention to detail, and it'll have close to the natural colors that you can show, and you'll use usually colored pencil to do that. And if there's texture or pattern, you want to mark that. And the parts are going to be correctly labeled, and I'll show you that I expect horizontal lines to show those labels. And don't use arrows, and don't crisscross your label lines. Do your best with spelling, and then you want to make sure that you've included the name of whatever you drew and the total magnification. All right, so here is an example of what somebody might have drawn if they looked in the microscope and this is what they saw. they've labeled the parts of this cell, but you can see that they've used arrowheads. Don't do that, just use a horizontal line. So don't use those arrowheads. We're going to look at some more examples. Okay, over here on the left, this is a correct scientific drawing. Actually, I'm going to back up also. This is observation of an animal cell under a microscope. It didn't specifically tell us what cell we were looking at, although maybe it's a generic animal cell, and it does give us our magnification, so that was important. Here, a correct scientific drawing is going to show what you see in your field of view. Each one of these labels is coming off of a horizontal line, and that line goes just directly to the part of the cell that is being labeled. So we have a vacuole up here. We'll talk about that in a couple lessons. Cytoplasm, the nucleus, and then this cell membrane on the outside. Also, this is correct because it has the scientific name of what we are drawing, and it has the magnification. Okay, when we look at this incorrect drawing, there are several things that are wrong here. Some of these lines are crossing, and we can't have any of our lines crossing. We also would like to have horizontal lines coming out to the side. It just makes things organized and makes it easier for people who are looking to see what you drew. Then down here, I don't have a listing of what it is that we drew or what the magnification was, so that's incorrect as well. Okay, more kind of incorrect drawings. We do have a bunch of cells, and that's okay, but when we labeled the nucleus here, we have an arrowhead, and that's not quite right. We don't want to use arrowheads, just a straight line. And then we have two lines crisscrossing over here. Also, it's best to go ahead and have all of your text be aligned, so this is a little too slanted. Okay, one other thing, nobody used any color on this, and we don't know what it was that was drawn, and we don't know the magnification. So in class, some of the classes were able to look at cork, which is a dead bark from a specific tree. All classes got to look at frog blood, and so let's talk about that frog blood. Okay. How do you know what you're looking at when you're looking at slides in the microscope? The slides are typically going to be labeled. There are some occasions where we won't have labels, but typically they're going to be labeled. So this is where they would be labeled usually. Okay, when you are filling in your sheet and getting ready to draw, you're going to go ahead and put your first and last name on your page and your period number if you see an indication for that or it's helpful anyway. Then on these bottom lines, just underneath the circle, which is going to represent your field of view, you're going to put your organism name and the magnification. All right, there are colored pencils near the microscope so that you can try to be accurate with your drawings. So when we look in our microscopes today, we're going to be looking at frog blood at 400 times normal magnification. And so you'll see on the frog blood that first we're going to focus in on low power. That's when we use our coarse adjustment knob. And then we're going to go to medium power and then to high power. And we should roughly see this kind of an image. Okay, we only need to draw 8 to 10 of our cells, and we're just going to label one of them. So the frog blood cells that we had do have a nucleus. Human blood cells do not have a nucleus. You can see that we've got a horizontal line listing the cell membrane, which is just the outer covering of the cell. And we have given ourselves more room, but we have a horizontal line leading to the label nucleus so that we are trying to be aligned. Okay. All right, so everybody's going to observe frog blood, draw 8 to 10 of the cells under high power, and just label the cell membrane and the nucleus on one of the cells. You're going to use your drawing checklist to make sure you're being accurate. All right, so some students are going to be working on microscopes, and then other students are going to be doing independent work. So with your independent work, you're going to learn about how scientists develop the cell theory. There is a specific cell theory that we subscribe to. And then we're going to connect our cell theory to the nature of science, which is something we did at the beginning of the year. All right, so I'll get you started on the independent work. And if you missed class, you're going to want to come in. You can do the independent work without being in the building, but you'll want to come into the classroom to do slide work. Okay, I'm just going to tell you that we talk about 5 different scientists in the independent work. The first guy is Hooke, that's his last name. This next one is going to be Leeuwenhoek. And then we have Schleiden and Schwann and Virchow. So that's how we're saying all of those names. Leeuwenhoek is particularly complicated, and sometimes you'll hear it called Leeuwenhoek. All right, so you'll go into Schoology Day 2 and work on the independent slides, and there's a video for you to watch. And then when you get a chance, come into the classroom so that you can look at the slides. All right, after you've done your independent research, you're going to come back here, hopefully you've done the independent work, and we'll have a quick review. Okay, so these guys, as they have worked together, and then of course many, many other scientists as well, they have come up with a theory about cells. So we know that all living things are made of 1 or more cells. All living things are made of 1 or more cells. Then part 2 is the cell is the most basic unit of life. Finally, number 3 says that all cells come from other cells. So we have a timeline that we're going to look at, and we'll look at this also in day 3, so you might get a little bit of this twice. I'm going to ask you to use this timeline, even if the video had some things in a slightly different order. In 1665, we have Robert Hooke, who is the first to see cork cells. That says cork just underneath my number there. Then in 1673, Anton van Leeuwenhoek observed living cells through a microscope. So Hooke just looked in the microscope and he saw something, and it looked kind of box-like to him. It looked like there were a whole bunch of little boxes side by side, and so he said they look like cells at the monastery. So he doesn't know what he's looking at, and it's not alive, it's actually dead, but he's looking at cork, and he says that they look like cells. A couple years later, Anton van Leeuwenhoek is also using the microscope. He managed to observe living cells, so he's credited with being the first guy to actually look at living cells. And then we jump up, I don't know, about 150 years or so, and Matthias Schleiden discovered that plants are made of cells. All plants are just made of cells. And nearly the same time, so this was in 1838, also we have a guy called Schwann, Theodor Schwann, who's working on microscopes as well. And some places you're going to see that he is listed as finding this out in 1838, and some places 1839. So here's why I want to say that we have Schleiden in 1838 and we have Schwann in 1839. Please use those dates. Okay, Schwann discovered that animals are all made of cells. And then finally, just, 15 years later or so, we have this guy called Virchow, Rudolf Virchow, and he discovered that all cells come from living cells. All right. These happen to be in alphabetical order, which if they are in chronological order, they're in alphabetical order. That's helpful to know that, if that helps you keep track of who did what. All right, so we also have some mnemonics as another thing to help you remember. And if you think about Schwann, he's the guy, it sounds like swan, right? And swans are animals. So Schwann is the guy who discovered that all animals are made of cells. And this one is a stretch, but Schleiden. If I'm sliding through the grass, grass is a plant. So Schleiden discovered that all plants are made of cells. Where's my there? And van Leeuwenhoek, what did he do? Well, he is credited with being first to see living cells. So you've got L for Leeuwenhoek and L for living cells. And then Virchow. If we say Virchow, sort of like Virchop, then we realize that Virchow is the guy who said that all cells come from other cells. He figured this out because he was watching live cells under the microscope, and he saw some divide. So he had one that divided into two, kind of like it was being chopped. All right, so make sure you've done those independent slides and watched the video on there. But we're going to come back to the idea of the nature of science. This is something we worked on very early in the year. And if we think about what we've just talked about, we know that scientific knowledge is durable. So since the 1600s, we've been talking about cells, right? And so we still talk about cells in a similar way. We also know that scientific knowledge can be subject to change. If one of those scientists believed that cells just kind of crystallize and appear on his slides, he's actually wrong. And we realize with Virchow that all cells, in fact, have to come from a pre-existing cell. They can't just appear out of nowhere. We also know that science is a complex social activity. I'm going to go back just so I can show you the timeline again. It's a social activity, and you can see that they work sort of together, right? So Hooke and Leeuwenhoek, they can share information. They are nearly in the same time frame. Definitely, Schleiden and Schwann are working together with microscopes at nearly the exact same time. And so they are able to listen to other scientists' findings and think about them and see if they agree. And Virchow, after hearing that some of the other scientists believed that cells can just appear, he kind of discounted that, disproved it a little, because he said, no, I was looking and I see that they divide and they make more cells just by splitting in half. All right, so we did our mnemonics already. And nature of science. So you have an exit ticket for this class day. And of course, remember, you can use your notes. And for now, you can always try a second time. All right, thanks for watching.